All right. So welcome to today's AP Biology overview session. Gary, I'm not sure if he's going to be able to make it because he's at the doctor's right now. He's also in Taiwan, so the schedule is they're 12 hours off. So we're just gonna see if he shows up. But if not, it'll just be me. Um, so yeah. I guess I'll get started with the presentation. So first of all, about me, just a few words. I took biology this year. I got a 5 on the exam, so hopefully that means I know what I'm doing. And I really enjoyed taking AP biology, and the reason why is because it really explained the whole field of biology to me. The way that they presented it in middle school and kind of in freshman biology, although I actually didn't take freshman biology. I'll talk about that in a sec. But in middle school and freshman biology, they kind of just threw terms at us and they didn't really explain what they were. They didn't tell us what ATP was. For me, back in middle school, ATP could have been magic and I wouldn't have known. And I wouldn't have questioned that because they didn't give me a reason not to. But you do learn what that is. Electron transport chain. That's a process that they taught you in middle school and freshman biology that existed. But again, you could have told me it's magic, and I would have no reason to believe otherwise because they didn't really explain what that is. In AP biology, they explain what that is. So no more mysteries about biology. It's not magic. It actually makes a lot of sense. Now, going into AP biology, what do you want? Math, biology, chemistry backgrounds, yes. Again, coming from me, I did not take freshman biology. I actually took the CDM for it. Would not recommend taking that CDM, but I did because everyone else did. And I was fine in AP biology. Although I didn't really have as much of a background in biology, I was fine because for the most part, they go into the stuff that that freshman biology covers. But they do explain, it's not like you need to know everything from freshman biology to take AP biology, but it is a prerequisite for a reason. Chemistry is the same way. Chemistry is a prerequisite for AP biology. And that's because you do kind of want to know what's going on with chemistry. But again, you don't really need to know exactly what's going on. It's very helpful to have a chemistry background though. And that's to understand stuff like pH, stuff like equilibrium and reactions. And math, finally, there's a few mathematical parts of AP biology. So probably, I guess, algebra 1, algebra 2 level math should be enough. But again, you don't really need to know that much. It's just very helpful. So starting now, I'm just going to go into the AP curriculum. And first of all, I guess I'll go into the AP big ideas. So there's 4 of them, but it's not like there's 4 units. The AP course actually, or AP recommends that they split it into, I think 9 units. My teacher at Chapel Hill High School split it into, I think, either 9 or 10, which weren't exactly the AP units, but they were pretty much aligned. So that's how the units are going to work. But with the 4 big ideas, I just wanted to put those out there because that's what AP defines as the most important ideas for the course. So the first one, evolution drives diversity and unity of life. That's where you kind of use math in biology. And it's kind of, it's like math, logic, game theory even. They've used that to prove evolution. So that's evolution. 2, biological systems use energy and molecular building blocks to grow, reproduce, and maintain dynamic homeostasis. That's where the chemistry comes in. And there's a bit of physics there too, but chemistry, it's mostly chemistry. And this is stuff that you'll learn in chemistry. How the energy and the molecules interact. So, and then the third one is living systems store, retrieve, transmit, and respond to information essential to life processes. This one, that's like chemistry, it's math. This is your genetics, basically. And then number 4, biological systems interact, and these systems and interactions exhibit complex properties. This is also chemistry and math. But those are the four big ideas. I'm going to go now specifically into the units that my teacher had, which pretty much align with the AP curriculum. And if you take it at Chapel Hill, this is how you're going to learn it. So the first unit is Introduction to Biology and Chemistry. Goes over the scientific method and also the fundamentals of observational and experimental science. So there's a few vocabulary terms here, and let me just turn on the laser pointer. You'll learn what these mean. Some of them you might already know, but it's good to have review anyway. And also you learn that biology is chemistry. Organic compounds are just chemicals. They're chemicals that are dissolved in water that interact with each other, and their properties determine how they interact and behave. Water, the biochemical solvent, you'll learn why that is. Although I'm pretty sure you learn about that in freshman biology. It's good to have a review. And finally, this protein structure determines protein function. That's actually really big. And the reason why is because pretty much everything in biology, everything in biology is about proteins. How do proteins fold? How do proteins interact with other compounds? That's going to determine how a living thing works. And this was like a really big, I guess, concept that our teacher emphasized. It was like really bold on his notes sheet. That protein structure determines protein function, and how you determine that protein structure is based off of chemistry. So yeah, that chemistry, that's basically how the protein interacts with water. I guess I can go into this because we're like 5 minutes in, so I'll go into this a bit more in detail. Basically, each protein is made of a chain of amino acids, and each amino acid has a R group and a side group, and basically that side group determines the chemical property of that amino acid. So some of them interact with water, don't interact with water. Basically, they're hydrophobic, and so those tend to fold away from the water into shapes. So let's say you have a tube, right? The inside of the tube is going to be mostly hydrophobic because that part is going to fold away from the water. Then there's charged ones that also kind of determines. So the charged ones, they kind of fold towards each other. Positive and negative charged amino acids, they fold towards each other. And then there's also hydrophilic ones, which are the ones that interact with water. And you'll learn about this more in the course, but there are a lot of areas where specifically or specific amino acids with specific properties are going to exist so that they can form a functional biological system. So the second unit is on cells. You'll learn that cells are just biological systems with different different parts that have different functions. Difference between eukaryotes and prokaryotes, that's just nucleus. But the organelles are also different. Prokaryotes, for example, do not have membrane-bound organelles that are seen in eukaryotes, but they do have organelles. So they told you in freshman biology, prokaryotes don't have organelles. They don't have eukaryote organelles, but they do have organelles. They do have areas with specific functions. Compartmentalization, that's big. Membrane-bound organelles stay apart from each other, and that lets them do what they do in the space that's most optimal. For example, a lysosome is very acidic, and that's because the enzymes in there that break stuff down only work in very acidic environments. And if a lysosome breaks, then those enzymes denature because they have a specific pH range where they work. And so that means if a lysosome breaks, it's not going to eat up your whole cell. So that's compartmentalization, and that's one example. Water potential, that determines how things are going to flow. So osmosis and diffusion, um, how water flows from one area to another, and then osmosis across a membrane. Water potential determines which direction that water flows in. Water flows from high potential to low potential. And a way to decrease water potential, for example, one way is to add solutes. So think about this in the way that they taught you in freshman biology. When you have a solute, the other side on one side of a membrane, and you have dilute water or maybe even pure water on another side. The solute is going to make the other side more concentrated, and water is going to try to fill the gap in concentration, and that's explained by water potential. So that's another demystifier in AP biology, how water potential determines how things flow, and it's all, at the end of the day, it's all like physics. It's all about equilibrium. Communication, so between cells and also inside a cell itself. There are receptors in cells, and each receptor triggers a response, and they do it by activating a chain of proteins that activates something. So each protein activates another protein, and they do that. They have this chain so that you can amplify a reaction. The protein doesn't turn off. Each protein that activates another protein, it doesn't turn off after it activates one protein. So basically it can activate a bunch of those proteins, which can activate even more of those proteins. So if one protein activates 10 of the next protein, each of the next protein activates 10 of the next, then boom, you get 100 times magnification. And that's why you have a lot of those steps. So you learn about communication, cell communication. There's also apoptosis, programmed cell death. You might have heard that term. You might remember that term from freshman biology. And that's basically just when a cell doesn't pass a checkpoint or when a cell detects that there's something wrong, if the DNA is irreparably damaged, then it's going to enter apoptosis and break up into components which can be easily processed. Because if a cell doesn't undergo apoptosis and instead like bursts, that can actually cause other cells around it to die, and then you have necrosis. You have a bunch of dead cells, and that's kind of nasty. Third unit is energy. This one is like really a lot of chemistry and physics in here. So living systems, obviously they use energy, and basically every reaction changes the energy of a system. There are unfavorable reactions which can't happen on their own, and there are favorable reactions which can happen on their own. And the reason why is because unfavorable reactions increase the energy of a system, while favorable reactions decrease the energy of a system. Or let's just say, the physics analogy here, when you're going up a hill, that reaction is unfavorable because you're increasing the energy, you're increasing your potential energy. And then when you go back down that hill, that's favorable. You're decreasing your energy. You're turning your potential energy into kinetic energy. And so you're decreasing the available energy. So basically, the whole point of this unit here is about free energy, but it's really, free energy is like available energy. How much energy is available to use for biological chemical reactions. And the reason why you use ATP, for example, is because the breaking up of ATP, the hydrolysis of ATP, is a very favorable reaction. You couple that to something that doesn't happen on its own usually. For example, active transport, let's say, when you're trying to move some solute against its concentration gradient, when you're trying to move it from a low concentration area to a high concentration area, you're going to need to use energy to do that. And you couple that with hydrolysis of ATP, which releases a lot of energy, and then you can get that unfavorable reaction to happen. I wrote here non-spontaneous versus spontaneous reactions. That just means unfavorable reactions don't happen unless, like, unfavorable reactions do happen, but they happen less in the unfavorable direction than the favorable direction. So reactions happen in both directions, but they happen much more in the favorable direction than the unfavorable direction. And this might all sound confusing because I'm trying to explain this in like 5 minutes. But when you take the class, you'll understand more about what this means. This is actually pretty much the biggest concept that you're going to have to learn, but it's not that hard to learn. So that's just a preview of energy. Enzymes make reactions more. They don't make them. They don't make them more unfavorable or favorable, but they do help catalyze reactions by making the initial, let's say, so each reaction has steps. Some of the steps might be very unfavorable, and the enzymes make those steps less unfavorable, or they break it into more favorable steps. Respiration and photosynthesis, those are cell processes. So respiration, you'll learn, is a favorable reaction. Basically, you're taking a high energy molecule and you're turning it into you're turning it into a bunch of you're turning it into low energy molecules. You get carbon dioxide out of respiration when you get sugars in. Electron transport, I'll put this in specifically because this one was one of those mysteries of freshman biology that they didn't really explain, but they do explain it in AP biology. They explain the specific reactions that happen. So basically, you get every time you have an electron carrier lose an electron, that carrier loses energy and to the next carrier, and that's the transport of electrons. And every time that electron is transported, that is a favorable reaction that powers an unfavorable reaction, which is pumping hydrogen ions across a membrane. And then those hydrogen ions, basically those now they've got to diffuse back to an area where they've got a low concentration, because that's the cells, or the mitochondria, have been pumping the hydrogen ions into an area of higher concentration, and they want to go back into a lower area, concentration area, but they have to go through a protein that creates ATP, and basically it turns like a rotor. It's really cool. And then photosynthesis also uses electron transport. Photosynthesis is an unfavorable reaction, but it's also powered by a very favorable reaction, which is the pigments transferring the electrons through sunlight. And so that's, or just through light in general. So basically, you get a lot of energy going in order to power this unfavorable reaction. And it's pretty complicated, but you'll learn about it in the class. Let me just get some water. So the fourth unit is on DNA and protein synthesis. This one is it's really nice because, again, you get to learn about the specifics of DNA replication. First of all, structure of DNA, that's pretty basic, but like how it was discovered, and then how the steps that they went through to kind of figure DNA out. It's really cool. So you'll learn that too. DNA replication. So basically, they'll teach you about the specific enzymes. There's a set of enzymes that replicate DNA, and they'll teach you each one of them. It sounds like a lot to remember because there's a lot of enzymes, but it's really not. You just have to kind of put two and two together, I guess. It's not that hard. Transcription, how you turn DNA into RNA. You get basically another set of enzymes that that gets RNA from a DNA strand. And translation, how you get from RNA to proteins. They told you in freshman biology that it happens on the ribosome, and that's true. But they tell you in AP more details. So basically, each RNA codon, so the set of three, can recruit a single, I guess, transfer RNA that's got a amino acid on it, and basically the ribosome moves down the RNA codon by codon, recruits those transfer RNAs, and then or was it tRNA? I forgot. There's a certain kind of RNA that's attached to the amino acids, and those are recruited by the ribosome to match with the RNA codon. But that's So that's translation. You'll learn about that more in detail in the class. Regulation, this one's interesting. So basically, um, every cell has the same DNA. Every cell in a system or in a biological in an organism has the same DNA, but not every cell is the same, and that's because they express different sections of the DNA, and they don't express different sections of the DNA. Certain cells have certain proteins that activate certain sections of DNA, for example. And you'll learn about that more in detail in the class. I'm saying that a lot, but again, this is like a basic overview. Regulation. What else? And there's also regulation of DNA replication. That too. And finally, viruses. Viruses, basically, they're self-replicating genetic material. So there are viruses that exist inside cell DNA. So they're actually sections of your DNA whose only purpose is to code for a protein that replicates that section of DNA, makes a cut somewhere inside the genome, and then puts that DNA in there. So basically, all those sections of your DNA are replicating themselves and putting them back in your DNA. So that's really interesting, and those actually drive mutation. Unit 5, cell cycle and genetics. Basically, you'll learn how cell division works. It's a very methodical process, and there's a bunch of checks to make sure that your cells don't improperly replicate. And if a cell doesn't pass each check, then it has to go back. And if it doesn't pass a check in a certain way, if it doesn't pass a check, or if it can't pass a check, then it's going to go through apoptosis. So that's cell division. Math, genetics. I'm reading off of it. So genetics, it's all about math and guesswork. So you'll learn that replication, DNA replication and sexual reproduction lead to genetic variety. DNA replication leads to variety because mutations happen. Mistakes are corrected most of the time, but sometimes they're not corrected, and that's what a mutation is. And sexual reproduction, obviously, you're combining different sets of genes, so that obviously leads to variety. Punnett squares, those are how you determine different phenotypes or different genotypes that determine different phenotypes. And then random mutation, you learn kind of how those happen, although they do go over those in freshman biology, but it's good to have a review. And then pedigrees. How do families stack up genetically? And in there's a lot of math, there's a lot of guesswork, a lot of logical thinking. It's pretty fun to go through a pedigree tree, in my opinion, to go and see, is this trait here, can it be recessive, for example? And there's a few characteristics that of recessive genotypes or recessive phenotypes. So that's the pedigree. That's a whole pedigree thing. Unit 6 is about evolution. First bullet point on the slide here is mathematical equilibrium, and that's because that's really what evolution is. Basically, evolution is trying to maximize fitness to the point where you reach an equilibrium where everything has the same fitness, has the same maximum fitness, or at least maximum fitness out of the group you came from. Hardy-Weinberg equilibrium, that's actually that's special because that assumes that that's about like population genetics. That assumes that there's no evolution, there's no outside sources of genetic material. So basically what you're saying is in Hardy-Weinberg equilibrium is that you've reached the point of maximum fitness and there's no evolution going on, and you can tell based on the different you can tell if a population is evolving based on whether or not it's in Hardy-Weinberg equilibrium. Speciation, that happens because of separation. It can happen because of so basically, okay, separation. Actually, I'll just go into separation in detail. Separation can be geographic. It can be reproductive. So geographic separation is where, you know, you can't get to another, when two populations split and they can't get to each other because for example there's a giant hole between them or something, or there's a mountain between them, and so that drives speciation because each population is going to evolve in a different direction. And they may evolve in the same direction simultaneously, but they cannot evolve together anymore, and so that triggers speciation. And then reproductive speciation, there's like basically for example there's like a certain type of fly that only mates inside I think it was hawthorn fruits or something. But then apples got introduced, and then some of them started mating in apples. So the hawthorn ones and the apple ones evolved differently. So that was an example of speciation, although I'm not too clear on the specifics now because it's been a while. And then finally, you'll learn about origin of life. How did life form? And it's kind of a mystery still, but there are hypotheses, and a lot of them make a lot of sense. So, for example, one hypothesis that's, as far as I can tell, widely accepted is that life originated from basically simple organic molecules that formed because of the high energy conditions of the young Earth, when you had volcanoes and basically no atmosphere, so you had meteors and stuff, and basically in that environment, you know, a lightning strike could turn, you know, nitrogen into an amino acid, and they actually did experiments that proved that can happen. And then how did those molecules kind of turn into life? They say maybe early nucleic acids, early RNA or DNA, or I think for the most part they think it's RNA. Early RNA molecules kind of, you know, they kind of had to figure out how to so they didn't figure out, but early RNA molecules that could replicate, well, they replicated and they outcompeted the other RNA molecules because there's a limited supply of RNA molecules. So basically, the strands that could replicate themselves would take up the other ones, and the other ones would degrade. It sounds really confusing, I know, and that's because I'm trying to go over this in 5 minutes. But you'll learn about that in biology, and it'll make a lot more sense. But it's really cool. Unit 7 is biotechnology. This one is like, I'd say one of my favorite units, and that's just because it really explains what's going on behind the scenes with what they're doing with the coronavirus research right now, and also with like how they're testing for the coronavirus, because we do learn what PCR is. In our class, we actually did a PCR lab where we took part of our genome and cloned a useless fragment of DNA to see if we had it or not. So basically, cloning, we go over cloning, gene analysis, and gene editing. Cloning, the two processes that we go over are bacterial plasmids. So they take a section of DNA, insert it into a bacterium, and then have the bacteria replicate those. And then there's PCR, which you can take genetic material, add polymerases. So those are the enzymes that replicate the DNA. You add some primers too to make sure that the DNA is replicated, because you need somewhere to start. And then after that, you add a bunch of nucleotides, and then the polymerases put them together. You'll have to continuously melt the DNA strands apart. So you have to separate them by heating them up. And then after that, you can cool them down, have the polymerases replicate the DNA, and then heat up, cool down, and so on. And that's how they can, for example, test for the coronavirus. They just test for that genetic material through that process. They replicate it, And if they can replicate it and find that there has been a lot of replication going on based on the coronavirus RNA or the coronavirus genetic material, then they will test positive for the coronavirus. Gene analysis, you have a few different processes. Gel electrophoresis, basically you're taking a section of DNA and you're running it through a gel, and how fast it goes through the gel depends on how long it is. So longer DNA sequences go through the gel slower because the gel is like a mesh, and the longer ones get trapped in it more. There's also DNA sequencing that uses gel electrophoresis to basically determine how the sequence is, and the process kind of goes the same, but instead you're PCRing a bunch of DNA, but you're randomly adding nucleotides at the end that cannot be. It's really complicated. I'm not sure if I can explain it in 5 minutes. If you want me to explain further, we can talk about it at the end. But I guess because I'm getting tongue-tied right now, I'm going to move on to genetic mapping. Genetic mapping is also genetic mapping is like that one's like math and guesswork and logic. Basically, it's a logic puzzle, and you'll learn how that works too. Gene editing, how they add genes. Again, you just have to be sure where to cut, and you have to use a certain type of enzyme to cut a gene in a certain place so that you can add a fragment of DNA to that gene. And then knocking out genes, you can use a few different methods. One kind of the most well-known, maybe not the most, but one very well-known one is CRISPR, and you'll learn how that works. Unit 8 is plants and diversity of life. So they kind of put it together, but plants is the main part, and then tree of life is another. You'll learn in this unit, it's actually not an AP unit anymore, but at least at Chapel Hill, they still do teach it. So plants, you'll learn how the characteristics of plants. For example, plants have 2 life cycles, one gametophyte life cycle and a sporophyte life cycle. You'll learn what that means in the class. And you'll also learn how plants behave. And then diversity of life, that's kind of just symbiosis, tree of life. Where does everything connect to each other? And that kind of builds off of the evolution unit, where you're now taking phylogenetic trees and you're putting together the relationships between different species. And then finally, the last unit is animals and ecology. You'll learn about the characteristics of animals, for example, that they all develop around a digestive tract. Everything from sea sponges to humans develop around a digestive tract. And then you get anatomy and physiology, self-regulation. So that's stuff like homeostasis. Why do we sweat when it's hot, and why do we shiver when it's cold? So they go over that. Some of it, again, review from middle school or freshman biology, but it's nice to have that. Human body systems, we actually didn't go through all of them this year because it's not an AP required unit anymore. And we also ran out of time. But human body systems, you'll learn about that more in detail. This year we went through reproductive system, excretory system, and digestive system. Sorry. And then finally, you learn about ecology. Just going through these terms here: dispersion, how are different populations dispersed across an area? Are they randomly, are there just random clumps? Are they all grouped together? Are they evenly spread out through an area? So that's dispersion. Population growth, there's some math in that. You get logistic equations and basically how the population growth works. Trophic levels, that's basically how much energy is available at a certain trophic level depends on how much energy is available at lower trophic levels. So that's all. Those are all concepts that they'll teach. Biogeochemical cycles. There's a carbon cycle where carbon from the atmosphere, so that's carbon dioxide, is fixed by organic means into organic carbon. That's stuff like your sugars. That's stuff like your organic molecules, your other organic molecules. And then there's also the nitrogen cycle, where atmospheric nitrogen is fixed by bacteria, and those are inside plants. So that's the nitrogen cycle. And then finally, biodiversity, why it's important, and how that kind of fits into how life is on Earth. That's the last topic that we covered in class. And basically, the big idea that came out of AP Biology this year for me was that there's really nothing special about biology. There's no pure biology, because everything, it boils down to either chemistry, physics, or math, and it really clears things up. How do biological systems work? Well, they work because that's how they work, and that's how chemistry works. That's how physics works, and that's how things logically work. So I think that's the big reason why to take AP Biology. It's because it's like enlightening. You learn that there's really, life is. I'm not going to say life isn't special, because life is kind of special, but life is, it follows the rules. It does things the way that they're supposed to, or it behaves the way that they're supposed to behave in accordance with the rest of science. And so that's something that they really don't teach you in freshman biology, but that's the special part about AP Biology. It's really where you can finally understand the whole point behind biology, and that's why you should take it. So now that I'm done with the content, I'm going to go into the exam format. So basically, the AP Biology exam is 3 hours. You get 90 minutes of multiple choice, which is a lot of time for 60 individual questions. You get sets of 4 to 5 questions. So basically, it's like a lot of the other AP exams. If you've seen any of the other seminars, or if you've taken other AP exams, you get a stimulus and you get a set of questions, and that's kind of how AP Biology works. There are some questions that are individual questions, but there are also some stimulus-based questions. 4 answer choices for each question. Most AP exams now only have A, B, C, D answer choices, but and biology is one of those. And the multiple choice is half of the total exam score. Free response is the second section. You basically get 6 questions. You get 6 questions. 2 of them are long. You have to take what they give you and make graphs and write long answers, and there's a lot of parts. And then there's 4 short ones. There's only like 4 parts for each of the short questions. And those ones are easier, I'd say, because you don't have to do the graphs and stuff. The answers kind of fold into each other. It's like the first part leads to the second part, leads to the third part, leads to the fourth part. And they do that for the long questions too, but the long question, they do it more for the short questions. And the free response is worth half of the total exam score. You're allowed to have a calculator. I'd say bring a graphing calculator if you're used to it, bring a scientific calculator if you're used to that. And you have an equation sheet, which I'm not going to take a look at right now, but that is available. You can just Google AP Biology equation sheet. And when you're taking practice exams, highly recommend, you pretty much have to take it with the equation sheet beside you. It doesn't matter if you don't use it, but you should know the equation sheet, and you should know where everything is on the equation sheet, so you don't have to deal with that on exam day. but that's really exam preparation. The next exam is next May so it's not for a long time. But that is a tip that I'm going to give out for exam preparation. First of all, take practice exams. There are a lot of them on AP Classroom, and you'll have access to AP Classroom if you're taking AP Biology or if you're taking the exam. There are a lot of them on AP Classroom, and there's also another one that's publicly available. I've linked that at the end of this slideshow. So this slideshow will be made available, and so will the video recording. So feel free to come back and review this slideshow when you're studying for the exam. And then the score distribution, Gary put that in there. Fives, 7.4% in 2021. I believe the 2022 ones came out, and I think it was higher. The 5% was higher. But AP Biology isn't like an exam that everyone aces. You know, it's not Calculus BC, it's not Physics C. It's not like, it doesn't have a very generous curve. But if you study by just taking practice exams, knowing your content, then you'll be fine. And you might have said, or you might have heard me say back there, know your content. This is another, I mean, it's kind of a no-brainer exam tip, but the better you are at the class, the better you are at the exam. If you can do well in the class, like get A's on all the tests without cheating, of course, then yeah, sure, you're probably going to do very well on the exam. And you know, if you can put the puzzle pieces together that make up the individual units, you'll probably do well on the exam. And a good way to tell, you know, how good you are is to take practice exams when you're studying for the exam at the end. Would I recommend taking practice exams before you go through all the content? Probably not. It's kind of a waste, I think, because you're not gonna be able to know how you are on exam day when you know everything. And there's a limited supply of practice tests out there. So use them wisely and take them. There will be time, a few weeks, to study for the exam after you get through all the content in the class. There's also this score calculator on albert.io. Take these score calculators with a grain of salt, but you can use these to grade your exams that you take, your practice exams that you take, and you'll be able to grade those practice exams because they have answer keys on each one of them. Tips for the multiple choice specifically: eliminate irrelevant information. Yeah, a lot of the AP questions give you unnecessary information. Although they're getting better with that, they're kind of doing it for us now, which is great. But yeah, that's a good way to not waste time. Underlining key terms or phrases. Honestly, I didn't do that on the exam, but if that helps you, then good for you. Do that. Use good time management. Don't dwell on questions. That's kind of for all multiple choice tests, but goes without saying, if you use a bunch of time on one question, you're not going to have enough time on other questions. And 75 seconds is basically 60 questions in 90 minutes. This is the average time spent on each one if you spend all your time on the questions. Skipping tougher questions and going back. Again, this is to make sure you don't miss a bunch of easy ones because you're stuck on a tough one. Each question is worth the same number of points. So yeah. Evaluate, describe, identify, explain. This is actually more of a free response tip, but just you know, being said here, evaluate and describe and explain. You have to go into detail, while identify you just name drop. And then third, make an educated guess. There's no guessing penalty, so if you don't know the answer, do your best. For the free responses, yeah, understanding the 4 big ideas is important because they're really testing you on those big ideas. Reviewing your labs and your data analysis just to make sure that you know how to interpret data like that, because you're going to have to be able to make graphs. You're going to have to be able to analyze data on the free response section, and knowing the question types. Again, recognizing the difference between those terms that AP uses a lot: evaluate, describe, identify, explain. Knowing the difference is important, and knowing the question types really comes down to whether or not you took the practice exams. So take the practice exams. That's the biggest tip that I've got for taking the AP biology exam. So that's it for the slideshow. Thanks for coming. Do you have any questions?
Not really.
Okay, then. Well, that's the end of the slideshow. Some materials for your reference: AP course and exam description. I don't really read these unless I'm trying to prepare to teach an AP class, but there is a lot of information in here about the AP course and exam, and there are also practice exam questions in here too. So that's another good resource to find practice questions. Also, AP released an official practice test. You can Google AP biology practice test, and you'll see this. This is from 2013, so it's not like aligned with the latest curriculum, I think, but it's pretty close. This is a very good resource, and I'm pretty sure I used this when I studied. So that's pretty much it for this slideshow. Thanks for coming, and let me just stop recording.